Hi and good day all. Today I would like to present my presentation on bioinformatics and artificial intelligence, a new revolution in biological research. Have you ever imagined trying to analyze your DNA sequence by reading and understanding billions of letters in them? Sounds impossible to a human brain, right? That is where bioinformatics and artificial intelligence make their way. In today's biological research, it is not just a scientist performing endless experiment in labs. It also powerful computers, algorithms, and machines learning models working together to solve complex biological problems. That's the purpose of bioinformatics. Starting from human genome to discovering life-saving drugs, the combination of bioinformatics and AI is revolutionizing the way we understand life and even making it easier than ever. In this video, we will explore how these technology work together and where they are being used and why they are shaping the human biology. Starting with the question, what is bioinformatics? Bioinformatics is the application of computational tools and analytical methods to capture, manage, and interpret biological data. It is not only involved the interpretation of biological information, but also leverages information technology to solve complex biological problems. At its core, bioinformatics facilitates the collection of analysis of data at the molecular level using computational techniques. Beyond the analysis, it also involves the storage of curated biological data in organized collection known as database. These databases are regularly updated to ensure the accuracy and validity of the stored data. Depending on the type of data, different databases are used. For example, protein sequence, genome sequence, and nucleotide sequence are stored in databases like UniProt, UCSC, Genome Browser, and DDBJ, and so on. Separately from those contain structural information and biological molecules, which is protein data bank. Although specialized databases exist for different types of biological data, they are often cross-referenced, and this interlinking ensures data integrations, consistency, and accuracy across different biological fields, enabling researchers to gain comprehensive insights. While on the other hand, AI or artificial intelligence can be defined as a field of computer science focused on the creating systems or machines that can perform tasks typically required human intelligence. These tasks includes learning, reasoning, problem solving, understanding language, and recognizing patterns. Among others, AI technologies aim to stimulate or replicate human cognitive abilities using algorithms, data, and computing power. Here comes a question. How does bioinformatics and AI contribute to the biological research? For bioinformatics, it plays a vital role in managing the vast data and ever-growing amount of biological data generated through experimental methods. It develops and maintains biological databases, organizing data in a searchable and accessible way for researchers. One of the most impressive capabilities of bioinformatics is its ability to predict 3D structures of biological molecules such as proteins, even when the structures are not available in existing databases, simply by using the sequences. Additionally, it also provides powerful tools and sequence level analysis, including alignment and comparison, enabling identification of conserved regions, functional sites, and evolutionary relationships. Moreover, it also facilitates the study of biological systems at molecular level, mapping interactions between molecules such as host pathogen dynamics, which is an essential step in understanding disease and developing new drugs. For contribution of AI, it is the driving innovation in biology by transforming traditional approaches and helping overcome the limitations. Using AI techniques like machine learning and deep learning patterns in complex biological data can be quickly recognized, enabling faster and more accurate analysis. It also facilitates the automation of experimental research, significantly accelerating processes such as drug discovery in the medical field. Moreover, AI-powered tools can analyze the vast amount of scientific literature and model biological system, making the research more efficient, insightful, and data driven. Significance of AI does not stop at research level; it is more deeper in applications. For instance, integration of AI has transformed agriculture field by introducing drone and robots to real-time monitoring crops and harvesting process, and even train autonomous robots to perform agriculture activities at faster rate than the traditional ways. Together, bioinformatics and AI form a synergetic approach that enhances accuracy, speed, and scalability in biological discoveries. This integration not only advances biological discoveries but also strengthens both AI and bioinformatics themselves. One of the greatest contribution of bioinformatics and AI to the biological research is AlphaFold by DeepMind. AlphaFold is basically a bioinformatics tool which is developed by integration of EMBL, EBI, and deep learning-based artificial intelligence system DeepMind, which is used to predict three-dimensional structure of protein by using amino acid sequence only. Now let's see the working principle of AlphaFold. First, it starts with retrieving sequence. Retrieves the amino acid sequence from known databases and it uses as an input for the AlphaFold. Next, AlphaFold performs the pairwise and multiple sequence alignment. Next, it proceeds to producing a representation of sequence analysis. After performing the pairwise and multiple sequence alignment, a presentation of analysis is sent to the Everformer deep learning module. Everformer, it produces an updated representation of pairwise and multiple sequence alignment which can be improved and reinterpreted. Using that updated representation, the transformer-based layer is processed and produce a spectral relationship with amino acid residues. At last, for the structure predictions, the transformer predicts the structure by transforming the abstract representation into 3D coordinates. The amino acids are considered as separate entity and rotations are predicted as well. To reiterative refinement, the predicted structure is refined by feeding the output into the models. Let's get into the real-life application of AlphaFold, finding pathogenicity of transmembrane 165 protein variant. TMEM165 is basically a gene that encodes for transmembrane protein. This protein is commonly found in Golgi apparatus, which shipping and packaging center of protein and lipid in cells, and function of this protein is deeply involved in the process like Golgi, homeostasis, and glycosylation, which is crucial process for protein folding and functions. When this gene is subjected to mutation, it may produce mutant variants which makes the expression and function impaired, leading to the congenital disorder of glycosylation. Currently, there is no cure for this disease, thus gaining a deeper understanding of the structure and function of mutated TMEM165 is essential for uncovering the underlying causes of the disease and advancing efforts in the drug development. For the contribution of AlphaFold, researchers utilized AlphaFold to predict the 3D structure of this protein as no experimental homologous structures was previously available. This predicted model was further refined using molecular dynamic stimulations in the presence of lipid, bilayer, and water, providing a more realistic structural representation. For the validity, these predicted structures by AlphaFold were validated by the experimental results such as Western blotting and immunostaining, which examined the expression and localization of mutated TMEM165 in the cell models. Also, the comparisons with the homologue in other species further validated the predicted structure and functional features, reinforcing AlphaFold's value in the studying rare genetic disorders like CDG. It also indicates that AlphaFold successfully identify the mutate regions in the structure. In conclusion, AI and bioinformatics have greatly accelerated progress in biological research from protein structure prediction to drug discovery. These technologies must be implemented with caution and oversight. AI should serve as a tool to support human intelligence, not to replace or override it.